Dear friends, this video about potato cultivation, breeding, biology and other related aspects. Potato, scientifically called as Solanum tuberosum, are a member of the order Solanaceae, which include tobacco and tomato, along with chili. It is however classified as a root crop, which is actually a misnomer, as potatoes are actually swollen underground stems or tubers. Tubers are swollen underground stems, which parts used to store food and genetic information. The chromosome number is 48. Origin is tropical South America. The floral formula is calyx 5, corolla 5, androecium 5 and gynoecium 2 and superior. Here this is a hermaphrodite flower. The economic importance of potato. Potato is said to be complete food as it contains carbohydrate, protein, vitamin B, C and minerals like phosphorus, calcium, iron required for body growth. Its calorific value is high, the richest source of starch. It produces more food per unit area than any cereal crops within short period. In India, it is used as vegetable alone or mixed with other vegetables. Potato is a basic ingredient of most of the snacks, curry and a major cash crop across the country. Coming to the area, production and productivity. In world, the total production is 368.1 million tons. China is 88.9 million tons. India 44.31 million tons. These two countries contribute 1/3 of total production of world's potato. Then Russia is the third country to contribute for potato cultivation, that is 30.2 million tons. In India, major potato producing states are Uttar Pradesh, West Bengal, Bihar, Punjab and Haryana. The total area in India is 2.02 million hectare, production 44.31 million ton and productivity 219.6 quintals per hectare. The major contributor is Punjab, that has production of 2.13 million tons. The nutritional constitution of potato: 100 grams of edible portion contains 74.70 water in grams, calories 87, carbohydrate 22.6 gram, sugar 0.9 gram, protein 1.6 gram, fiber 0.4 gram, fat 0.1 gram, minerals 0.6, calcium in mg 7.7, copper 0.15 mg, iron 0.75 mg, magnesium 24.2 mg, phosphorus 40.3 mg, potassium 568 mg, sodium 6.5 mg, vitamin C 14 to 25 mg, thiamine 0.18 mg, riboflavin 0.01 to 0.07 mg, niacin 0.4 to 3.1 mg, total folate 5 to 35 mg and pyridoxine 0.13 to 0.25 mg. The origin and distribution of potato. This is native of tropical South American region. Believed that the cultivated potato originated from its wild ancestors near the Lake Tropica basin in Peru, Bolivia region in high mountains. The potato was introduced in India from Europe in early 17th century. The classification of cultivated potato. Three are artificial groups based on wild. These are highly diverse. Then cultivated indigenous. They belong to Andes and southern Chile region, and modern cultivars. The ploidy became a taxonomic trait for cultivated potato. Andean fields contain mixture of land races and hybridization with wild potato. most accepted taxonomic treatment is Solanum ajanhuri, that is diploid, Solanum chaucha 36 triploid, Solanum curtilobum 60, that is pentaploid, Solanum zuzepzuki, that is diploid 36, Fureja 24. Here Fureja stenototum and curtilobum are the major wild species or the wild relatives which mainly contribute for the disease resistance or other quality traits. In Solanum tuberosum there are two species, Solanum tuberosum species tuberosum, that is cultivated potato, and Andigenum, that is also cultivated potato in Andes region. The morphological support for previous classification, in the previous slide was a old classification and the morphological support was poor for that, and in this slide the newer group is given, that is Solanum tuberosum, that is the only cultivated species because reticulate origin, multiple origin and continuous hybridization. The groups are classified as Ajanhori group, Chaucha group, Curtilobum group, like that. Coming to taxonomical classification, it is largest genus in Solanaceae family with the estimated 1400 species. Potato belong to kingdom Plantae, phylum Anthophyta, division Magnoliophyta, class Magnoliopsida, subclass Asteridae, order Solanales, family Solanaceae. The different growth stages of potato. The first stage is sprouting, initiation and emergence between 15 to 30 days of planting. The total duration is around 120 days from where germination to emergence or tuber final development takes place. The second stage is stolon initiation occur simultaneously than the emergence. In the third stage, tuber initiation between 15 to and 30 days of emergence. The fourth stage is tubers filling between 45 and 90 days of emergence. Then maturity and harvest between 90 and 120 days. The crop closing period is between 35 to 40 days of emergence. Coming to the botany, potato stolons are lateral shoots actually from most basal nodes below soil level. Typically they are diageographic shoots with enlarged or elongated internodes hooked at tip. Tubers develop from the subapical regions of stolons. The tuber formation includes two processes, one is stolon formation and second is tuberization of stolon tips. Stolon formation usually begin at the lower nodes and progress acropetally. The stems of potato, it is consisted of runner and tuber and stem. It grows as a single stem for few times and then divided into branches. 1 to 9 suckers are emerged in one plant. Stems divided into 3 categories based on height of the plant and flowering. Short is less than 45 cm, moderate is 45 to 60 cm and tall is greater than 60 cm. The buds, there are mainly two parts in bud. Growth of buds is affected by storage conditions of seed potato. Basal parts are at under the soil and therefore both runners and tubers are developed. Leaf, potato has compound leaves. It has prominent midrib and it is also divided into leaflets. Leaf is important to identify a variety. Flower, it's an inflorescence axis first divided into two white and purple color petals are closely arranged and made a corolla. The floral parts are regular in their arrangement. The flowers are bisexual, that is the stamens or male parts and pistil female part are on the same flower. Flower consists of four basic whorls: calyx, corolla, stamens and pistil. The whorl's calyx consists of 5 sepals that are partly joined at the base, forming a bell-shaped structure below the corolla. Shape and size of the lobes or free ends of the sepals vary according to cultivar. The calyx color may be green or partially or totally pigmented. Corolla consists of 5 petals which are joined at the base by a short corolla tube, each lobe and in a triangular point. Outline of the corolla is generally round. Some primitive cultivars have pentagonal or star-like corolla. Corolla color may be either white, light blue, blue, red and purple with different tones and intensities. The androecium consists of 5 stamens that are alternate with petals. Stamen is composed of anther and filament that are joined in the corolla tube, which can be seen in the photograph given below. Anthers are generally fused in the conical column enclosing pistils. In some cultivars, they may be spread apart loosely. The color of anthers varies from light yellow to deep orange. Pollen grains are shed through pores at anther tips. Gynoecium or female element of flower consists of single pistil which is composed of ovary, style and stigma. Ovary is superior. The sepals, petals and stamens are attached at the receptacle just below the ovary, that is nothing but superior ovary. In transverse section, ovary shows 2 cavities where generally numerous ovules are arranged along the periphery of placenta. This is a typical colored flower of potato where the fused anthers, stigma protruding outside and pollen dropping from the holes are seen in this picture. Coming to tuber, it's a modernized stem or expanded runner. It has eyes same as the general stem. One or more buds may be in one eye. Tubers not only store food as well as act as a planting material. Potato tuber is modified stem with shortened axis and rather poorly developed leaves. The inflorescence is cymose. Vegetative shoot is sympodium. Each portion terminates in the inflorescence. The tubers are also used in all genetic studies as I have discussed in the previous slides. The cytology or cytogenetics of potato. It has basic chromosome number 12, form diploid to hexaploid species. About 75% of species are diploid while 15% species are tetraploids. Triploids and pentaploids are highly sterile and maintained by vegetative propagation. Solanum stenototum, Solanum forager and Ajanhui are cultivated diploids of which former 2 are sexually fertile while later one is less fertile and yet bred true. Solanum chaucha and Jujepejuki are cultivated triploids are less sterile. Cultivated tetraploid species Tuberosum species tuberosum and Tuberosum species andagina are fertile. Solanum curtilobum is pentaploid. Regular meiosis have been observed in diploid, allotetraploid and allohexaploid. Nearly all the diploid species are self-incompatible while all the tetraploids and hexaploids are self-compatible. The genetics of potato: skin color, gene is digenic and gene action is complementary. For flesh color, genes are monogenic and the gene action is deep yellow or white. For stolen length, number of genes are monogenic, short or long. Position of eye, that is also monogenically controlled. That means apart from skin color, other traits are controlled by monogene or single gene. Coming to the breeding objectives, High tuber yield is a first and foremost breeding objective, followed by earliness, photoperiod insensitivity, responsiveness to fertilizer, better keeping quality, maybe for resistance, tolerance against shrinkage, rottage, etc. Better quality tubers, resistant to late blight, early blight, charcoal rot, common scab, and bacterial wilt. The different breeding methods, though it is a self-pollinated crop but vegetatively propagated. The cultivated tetraploid varieties are highly heterozygous. Most of them are also pollen sterile. Selfing or inbreeding in potato leads to loss of vigor of the progeny and non-flowering. Hence, conventional method or pure line method of breeding is generally not preferred or practiced. Next method is hybridization and selection. In hybridization, crosses are made between selected parents. Hybridization can be between intervarietal or interspecific since yield and most of the desirable characters are polygenic in nature. The parents for hybridization are generally selected on the basis of their combining ability. Being vegetatively propagated, breeders take advantage of selection and multiplying genetically identical individuals in succeeding generations. Backcross method. Cultivated potato doesn't possess resistance to most of diseases and pests. Resistant genes are mostly found scattered in wild and semi-cultivated species available in the center of origin of diversity in Andes region of Southern America. In this method of hybridization, it is done between cultivated and wild or semi-cultivated species with the aim of transferring specific characters like resistance to diseases and pests. It is followed by repeated backcrossing, keeping cultivated type as recurrent parent. Selection is practiced in successive backcross generations for the character to be retained from the wild species. However, transfer of the resistant gene from wild species into cultivated potato is a difficult task. Next is hybridization technique. Here, potato naturally flower under cool climate and long day conditions of more than 15 hours light. Such conditions are available during long summer days when potatoes are grown in hills. Hills are therefore ideal for hybridization work. Potato flowers are hermaphrodite and therefore emasculation is done in selected female parent mostly in the evening. The biotechnological aspects. Tissue culture technology widely used in production of disease-free and quality planting material. CPRI Shimla, that is Central Potato Research Institute, has developed transgenics using CryLab and S-motion gene for resistance against potato tuber moth and late blight disease. CPRI, in collaboration with JNU New Delhi, developed 8 Indian varieties using AML-MAL gene for increase in total protein content. Coming to the management practices or agronomic practices, the first and foremost is land preparation. Soil is turned up to 30 to 40 centimeter depth and make into fine texture. Then lime is added when soil is acidic in first land preparation in order to avoid the soil-borne diseases initially. Soil suitability. Ideal soil is a deep, well-drained, medium heavy loam with a few as stones as possible, because the loose soil helps in spreading or increasing the size of tubers. The high pH should be avoided and pH of not lower than 5 should be used. The optimum pH is 5 to 5.5. South facing, gently sloping fields are best for first early years. The soil must be frost free, so coastal area are very suitable. However, main crop potatoes can be grown in most areas. Then making ridges and furrows. Drain are prepared. Spacing between two drains is 60 cm and depth is 20 to 25 cm. Ridges and furrows are prepared in contour lines to reduce soil erosion in steep lands. Length of the ridge can be decided based on the location of the field and easiness of maintenance. Seed selection. Generally, whole tubers with small and uniform size are used in wide multiplication and hybrid seed production. Tuber should be true to type or genetically pure. It should be free from infestation of pest and disease like tuber moth, bacterial wilt, and virus. It should be healthy, well matured, and uniform in size. Each tuber should have 2 to 3 sprouted buds. It requires a rest period of about 2.5 to 3 months before they can be used for seed purpose. Medium to big size tuber should be used for planting. The seed rate recommended is for a whole tuber 15 to 22.5 quintals per hectare and for cut tubers 15 to 20 quintals per hectare. Tubers having 30 to 50 gram weight are the most economical and give highest yield. While cutting the tubers, care should be taken that each piece has 2 to 3 eyes and weight at least 25 grams. Coming to the factors considered while choosing a variety, farmers has to see the consumer preference. He has to see whether people like the particular variety or a processing plant is nearby his location. High yielding or eating quality, maturity, keeping quality, disease resistance, seed availability are the other factors which farmer has to be focused. Field planting. Selected seed potatoes are put on furrows as suckers are in upward direction. Then the seed potatoes are covered with soil of the ridges. Spacing and seed potato requirement. Inter-row spacing is 60 cm and intra-row 25 cm. Therefore, 2 to 2.5 tons of seed potato are required for 1 acre. The fertilizer requirement. For different soil types, the fertilizer requirement differs. Organic matter like cattle manure for delayed growing 0 to 12 tons per hectare, poultry manure 5 to 6 tons per hectare, organic manure 25 to 30 tons per hectare, or neem cake 3 to 5 tons per hectare. One among these any organic matter can be used. Then chemical fertilizers. The typical application rate is 10 is to 20 or 7 is to 6 is to 17. Sulfate of potash are ideal for potatoes. Fertilizer should be applied prior to sowing. The NPK dose for alluvial soils is 180 is to 80 is to 110. For black soils, 115 is to 45 is to 50. For red soils, 120 is to 115 is to 135, and for hill soils, 115 is to 135 is to 95. Earthing up. This is an important post-sowing cultivation of the potato crop. When the plant is 15 to 20 centimeter height, a ridge is used to bank up more soil around the stem. That is nothing but earthing up. This is important because to keep soils loose, prevent greening of potatoes, stop annual weeds and from germination, that can help to prevent diseases like blight and can help to increase the yield. Irrigation. Potato can withstand drought condition, but adequate amount of irrigation is required for better growth and yield. However, limitation of irrigation is required when harvesting. Irrigation should be done to wash away the dew of the leaves. Critical stages are most recommended stages of irrigation. Weed control. Glyphosate is used to destroy weeds at land preparation. Weeding is not commonly practiced in potato cultivation because it may disturb the tubers, because tubers are the commercial products or consumable products. So in order to avoid the poisoning or adverse effects, the chemical weedicide use is less preferred. Coming to post-harvest practices. When crop is ready to harvest, it is 80%, leaves become yellowish in color. Removing the shoots from the ground level enhance the maturity in upcountry areas like hilly areas. Tubers are suitable to harvest if peel is not loosened when compressed by fingers. Damaged and undamaged tubers are graded after harvesting. This is harvesting and packaging management. Harvesting of potato is done before temperature rises above 30 degrees Celsius. Harvesting is done manually with the help of spade or bullock driven single row plow or mechanically with the help of potato digger. Average yield of early maturing varieties is 20 tons per hectare, while that of late maturing is 37 tons per hectare. The small size tubers should be kept at seed tubers. The harvesting care should be taken during the crop harvest, like always harvest in dry weather. Harvest the crop after 15 to 20 days of dehalming. Avoid bruising and skinning of tubers, otherwise tubers become susceptible to rot disease. Harvest the crop after 10 to 15 days of haulm cutting. Potatoes are harvested from May to early June on depending variety and part of the country. Main crop potatoes are harvested in late September or October when the tubers are fully grown and mature, that is kharif potato. Drying. The cares should be taken during drying. Always dry the harvested tubers quickly to remove excess moisture from the surface of tubers for improving their keeping quality. Shade drying to avoid greening of tubers from hot sunshine. Always dry the harvested tuber in storage shed. Exposed to sun causes greening of potatoes. Do not store the tubers immediately if they are exposed to rain after harvest. Here I just want to focus on the greening problem. Greening is a chemical substance which gives adverse effect on human health. So greening is majorly avoided when the potatoes are consumed for home purpose. Delay in harvest will spoil the quality due to high temperature. Harvested tubers should be kept in cool place for about 15 days for curing. Tubers should be graded before transporting to cold store. Small size tubers should be kept as seed tubers. Curing. In this, always follow curing process at 25 degrees centigrade with 95% relative humidity. For optimum suberization, curing is essential for healing the wounds of tubers resulting from cutting and bruising during harvesting. Next is sorting. All the damaged and diseased tubers should be removed during sorting. Pest and insect problem may be avoided by this. In hills, tubers are spread in well ventilated rooms. Later, cut, damaged and rotten tubers are discarded and good tubers are graded into four size, that is small 25 gram, medium 25 to 50 gram, large 50 to 75 gram and extra large above 75 gram. Thoroughly clean and disinfect storage facilities and all grading and handling equipments after planting is completed and gain prior to harvest. Cure potato at 13 to 16 degrees Celsius and relative humidity of 90 to 95% for 8 to 15 days to heal wounds and bruises. Seed potato should be stored at temperature of 3 to 5 degrees Celsius with 90 to 95% humidity for long-term storage. Restrict the entry of footwears inside the storage chambers to avoid contamination. Grading. There are two grades, grade A and grade B. The round sized grade A is 50 to 80 mm and grade B is 30 to 50 mm. Long means 45 to 70 mm is grade A and 30 to 45 mm grade B. Seed treatment and packaging. Each variety of seed must be stored separately. Sprout inhibitors should not be applied in the field or in the storage because residues may evaporate and may re-establish in the new storage and may hinder germination in the future. After grading, the tubers are washed with 1% chlorosin solution followed by rinsing in water and dipping in 3% boric acid for 20 minutes. After treatment, the tubers are dried in shade and packed in gunny bags, then labeled and sealed and kept in cool storage. The storing ware building which store the potato must be 2 to 4 degrees Celsius temperature and 75 to 80% relative humidity. Potatoes stored at less than 0 degrees Celsius suffer from internal breakdown known as black heart disease. This is a physiological disorder. Low temperature prevents sprouting and rotting. That should be leak proof, frost proof. Potatoes are also covered with a layer of straw to protect against frost, well ventilated to prevent sprouting and rotting. In planes, the cured tubers after grading are stored in high relative humidity to reduce weight loss in tubers. Big cold storage or stores for potato are established in main producing states by APHID and other agencies. In indigenous method, seed tubers are stored in a single layer on sand. Frequent examination is necessary to discard tubers showing symptoms of rotting. Tubers are also stored in pit method. Here, pits of 60 to 75 mm depth, 240 cm length and 90 cm width are made in cool shady places. Water is sprinkled inside the pit to cool it. After 2 days, pits are lined from inside with neem leaves, dry grass or sugarcane trash. Bamboo chimneys of 1.2 to 1.5 meter length are placed inside the pit of 1.2 to 1.5 meter apart for escaping moisture controlled during initial stages and is managed by hoeing and weeding at 1 month after planting, followed by fertilizer application and earthing up. The damaged and diseased tubers are sorted out prior to storage in sorting process. Potato is stored in the layers less than 2 tuber thickness in the dark room with better ventilation. Coming to major diseases and pests of potato. In diseases, leaf roll. This is virus transmitted by aphids. Symptoms are rolled leaves and stunted growth. Control is control using certified seed and aphids. Leaf mosaic disease is transmitted by aphids that cause mottled leaves, stunted growth. For this also certified seed is recommended. Then black leg, Erwinia keratoria is the causal organism that is leads to blackening of lower parts of stems, blackening of stored tubers. Gangrene of potato, that is due to soil-borne fungus, Phoma exigua. Dark colored depressions on stored tubers, secondary infection by bacteria. For this, avoid damage at harvest and apply chemicals if necessary. Common scab. This is a soil-borne bacterium caused by Streptomyces scabies. Dark superficial scabs on tubers which affect selling quality. Leaf roll. This is also a viral disease caused by symptoms are rolled leaves, stunted growth. Leaf mosaic. This is also a viral disease where mottled leaves and stunted growth. Black leg is a bacterial disease. In this, lower stem becomes black and stored tubers become black. Coming to the major sources for disease resistance. These are wild sources. For major virus, that is potato virus X, Solanum aculae, Solanum burthouti, tuberosum species Andegina stoloniferum are the major sources. For potato virus Y, Solanum foragea, Solanum demisum, Solanum stoloniferum. Then for leaf blight, Solanum demisum, Solanum bartholdi, Solanum stoloniferum. Early blight, Solanum saltans. Wart, Solanum aculae and Solanum barthoti. Common scab, Solanum checons, Solanum tuberosum species Andegina. For bacterial wilt, Solanum chocoens, Solanum microdontum. Root knot nematode, Solanum spagazzini, Solanum verni. Cyst nematode, Solanum tuberosum subspecies Andegina, bartholdi or verni. For aphids, bartholdi. For frost, acole, ajohori, curtilobum and stoloniferum. For heat tolerance, chocoens, commersoni, anamelocalyx. For high protein content, Solanum foragea, non-dormant type, chelcoens and foragea, Colorado beetle, stoloniferum. Coming to the major pests, first one is wireworms. These are most likely to be numerous in first 2 years after a lay. Same species also attack barley. They attack the tubers, eating into them and lowering tuber quality. To control them, insecticides like aldrin is worked in the soil before planting, gives a good control. Aphids like saps of the growing plant responsible for spreading the disease, where number more than 3% or 3 per plant, use aphidicide or insecticides. Then slugs. This can be effectively controlled by pellets or pellet broadcasting over the soil. They cause troublesome, especially after wet summer and where soil is rich in organic matter. Eelworm. This affects crop root system. Nematicides or LD-carp granules can be applied. Coming to weed control, crops are sprayed with a contact herbicide when 1/4 of plants have appeared over the soil. A contact or residual herbicide kills all the plant tissue comes in contact with, if it will come in contact. The rest of the plant recovers. This allows for weeds to be controlled until shading occurs, which naturally prevents weeds. Shading refers to the time when plants meet across ridges. Earthing up also prevents weeds. Coming to the different propagation methods. There are 2 types of propagation methods of potato. One is sexual and one is asexual. Sexual is true potato seed and asexual is micropropagation, meristem culture, seed potato and tuber indexing. Among all these methods, in India from ancient period, seed potato and tuber indexing are the major processes which were carried out, but nowadays true potato seeds are coming up or taking a wide area, and it is helping the farmers in order to overcome the viral diseases. Coming to the first method in sexual propagation, that is micropropagation. Here first the microplants are grown in the culture media in vitro, then culturing in the liquid media in conical flasks, then microtubers are produced. Microtubers of 50 to 60% survival are screened in greenhouse, then mini tubers are obtained from there, then field multiplication for one time, twice, then breeder seed is produced. And micropropagation is the best method in order to develop breeder seed because it is devoid of all sort of diseases and other contaminations, and uniformity is also expected. The systems of seed production. First one is nucleus seed, that is the basic seed in seed production system. In this, stage 1, stage 2, stage 3 and stage 4, that means 4 years will be taken in order to multiply the nucleus seed and breeder seed. The second is foundation seed. For foundation seed 1, it is 5th year, and for foundation seed 2, it is 6th year. Means if we will divide to 2 stages for nucleus seed, 1st and 2nd year, breeder 3rd and 4th year, foundation 5th and 6th year, and for certified 7th, 8th and 9th year. Certified pure means it is 7th year, then certified seed 1 and 2 means it is 8th and 9th year. The different stages in different seeds is only when the demand is more and supply is less. Coming to seed potato concept. Potato tubers of definite size are used for further multiplication is nothing but seed potato, that is seed or potato is selected for the seed purpose. It should be free from all viral diseases, should not have been grown in areas where wart, cyst, nematode and other quarantine diseases are endemic. It should be either free from soil and tuber-borne diseases to carry them within permissible limits. It should be of proper physiological age. Next is true potato seeds. This is a potato obtained by or seed obtained by sexual process. There are 3 ways of producing true potato seeds. One is direct seeding of true potato seeds in the field, then transplanting true potato seeds derived seedlings, or planting seedling tubers raised from true potato seeds. The problems in true potato seed growing is low germination percentage, heavy weed problem, slow growth in of plants, dormancy period of 4 to 6 months, and minimum isolation distance of 50 meters. Coming to the importance of true potato seeds. The true potato seed is obtained by crossing 2 parental lines of potato, a substitute of traditional seed tubers. 100 grams of seed is sufficient to cover 1 hectare area instead of planting 2 to 2.5 tons per hectare of potato seed tuber. Being hybrid, TPS is capable of giving more production. Absolutely disease-free seed material. No cold storage facility is required for storing TPS. Practically no cost is involved for transportation, unlike seed tubers. Comparatively more resistant to pest and disease. Net profit is more as cost of cultivation is less, and also as the per hectare production is more. Non-availability of quality seed tubers, high seed cost, viral infiltration in seed tubers causing degeneration of seed stalks, and problems of long distance transport of seeds from seed producing area have led to the development of true potato seed technology for crop production. This is a picture of true potato seeds. The size is quite small and transportation or packaging is very easy. Coming to a concept of shift from hills to plains. Seed production shifted from the hills to the northern plains to cater the large demand of seeds. Hill seed may not be right physiological age to use in the plains. Hill soils may carry soil-borne pathogens due to continuous potato production, which often not found in the plains. Long distance transport area in hill is insufficient for seed production. Coming to the process of growing or agronomic practices into potato seeds. Seeds are sown on 0.5 cm depth in raised nursery beds of 6 inches or 15 cm, prepared to good tilt with finely powdered dry cow dung in rows at 10 cm apart and provide shade. Water with the fine rose can. Then apply foliar spray of 0.01% urea solution from 15 days after sowing on alternate days till the seedlings are ready to transplanting after 25 to 28 days with 3 to 4 leaf stage. Care should be taken against termite and damping off. Cultivation in the main field. To cultivate in the main field, prepare main field to a good tilt. After leveling, apply fertilizer at the rate of 75 is to 100 is to 150 kg per hectare. Make ridges of 15 cm height and furrow of 50 to 60 cm apart in east-west direction. Irrigate the furrows to 7.5 cm height. Transplant the seedlings on the next day on the northern side of ridges at half the height at 15 cm apart. On 35th day, apply 75 kg of nitrogen per hectare after weeding and earthing up is to be done in such a way that the plants come up on top of the earth. Provide irrigation as and when required. The tuberlets. Small tubers up to 20 gram size use as seed tuber. The basic material for true potato seed is seed tuber. Then the seed tuber requirement could be brought down to 1/3 by using tuberlets. Package of practices for production of tuberlets using true potato seeds. At present, there are two methods of practice for producing seedling tubers in bed. One is single row method and one is double row method. In single row method, prepare beds of 15 cm height, 1 meter width and according to convenient length at 0.75 cm apart. Bring to good tilt, mixing with finely powdered, well rotten dried cow dung. Apply urea, single superphosphate and MOP at the rate of 20 grams, 60 grams and 25 grams respectively as basal dose per square meter. Sow 2 to 3 seeds per hole at 0.5 cm. The depth should be 22 into 5 cm. Provide shade to avoid scorching sun and irrigate the beds with fine rose can as per necessary. Earthing up with the mixture of finely prepared soil and cow dung along with urea at the rate of 5 grams per square meter at 30th, 45th and 60th day. Cut the haulms at 85th day. During the whole production period, need base spraying at PPC should be undertaken. Treat the tuberlets with 3% boric acid and store in cold storage for next year after proper drying in shade. In the double row method, preparation of field and other operations as same as single row except sowing of seeds. In double row, seeds are sown 4 cm apart in a line and row to row distance of 10 cm. In between two double row distance is 30 cm. Top dressing with 5 grams. Urea per square meter at 30th, 45th, 60th day followed by earthing up as practiced in normal crops so that two lines can be covered by a single furrow. Profuse flowering in the parental line is essential for true potato seed production. Potato being a temperate crop needs cool climate and longer photo period for flowering to occur. At high altitude, potato is grown during long summer days and the plants produce flowers under natural conditions. Several characters are important for commercial production of hybrid true potato seeds and its utilization for growing were potato crops. These include high berry and seed set in the parents, which facilitates production of more seeds per plant, seed germination, seedling survival on transplanting and uniformity of production. Coming to the seed standards for potato seed production. Weight of mini tuber should be 1 gram, physical purity 98%, germination or sprouting purity minimum germination is 90%, varietal purity 99%, seed-borne virus 0.01% maximum, isolation distance for foundation seed from other varieties 5 meter and certified also same, field of the same variety not confirmed by the varietal purity requirements for certification. Foundation seed is 5 meter and certified is 5 meter. Coming to components of seed plot technique, in Punjab and Haryana planting date is October 7, dehalming on December 31st and harvest January 15. In northwestern and central plains, planting date is October 15, dehalming January 10 and harvest is Jan 15. In eastern UP and Madhya Pradesh, planting is in October 31st, dehalming January 15 and harvest during January 31st. Bihar and West Bengal, November 17 is planting date, dehalming January 20 and harvest on January 31st. Systemic granular insecticide at planting and earthing up are applied. Removal of off types and diseased plants. 1 to 2 sprays of systemic insecticide, metalaxyl and mancozeb in December and January, and crop rotation for 2 to 3 years. Tuber treatment with boric acid are highly recommended by CPRI Shimla. Coming to the impact of seed plot technique, Central Potato Research Institute produce 2,550 tons of breeder seeds per year. It gives 2,000 tons to NSC or state seed certification agencies. 2,000 ton breeder seed is produced at CPRI. The multiplication rate is 6 times for foundation seed and 12,000 foundation seed stage 1 is produced. 72,000 tons of foundation seed is produced in stage 2 and in stage 1 of certified seed 4,32,000 tons or 0.43 million tons of seeds are produced. The seed requirement of the country is 5.97 million ton. Solution is farmer can grow own seed with suitable agronomic strategies free from virus and disease. This is a boon from seed plot technique. This saves many US million dollars annually on seed import, and India is the only Asian country with a well-established scientific seed production program. Coming to certified seed production, these are produced accordance with certain procedures like first selection, then initial propagation and further propagation. The first process is selection. Potatoes to be used as seed must come from fields that are completely free from potato root nematode and must also be free from infestation. They are also strictly tested for 3 principal diseases, that is potato virus Y, leaf roll and leaf mosaic disease, and these seeds must come from the fields free of potato root nematode. Second step is initial propagation. The selected potatoes are propagated using 2 methods. They are grown under strict growing conditions in high elevation by windy nucleus plots or else using micropropagation techniques in lab. These plots are strictly controlled and are isolated from one another. The growing vegetation is regularly tested for viral infection. In the third step, that is further propagation, once enough seed is available, it is distributed to selected growers nationwide and propagated further in isolation and supervised. They are continuously tested for viral and other diseases. This final propagation are sold as certified seed to farmers. These seeds are not true seeds but immature asexual tubers or clones of parent plant. The quality of new plants will be identical to parent. Seed is grown in 1 to 5 rotation system. The advantages and disadvantages of certified seed. Advantages means seed has high % germination, crop has better rate of establishment, seed has higher yield, seed is true to type, fewer pest and disease, easier to market the crop. Disadvantages are quite expensive, isolation required, non-availability of the seed, transportation, lack of awareness, requirement of non-infested field from pathogens. Coming to the seed certification standards for seed potato. The classification of seeds is hill seeds and plain seeds. The hill seeds means 2500 meter above sea level, and plain seeds were low aphid infestation during crop growing season. The land requirement is not infested with wart, cyst forming nematode, brown rot, minimum within the previous 3 years, or no common scab. Third is field inspection. Minimum 4 inspections are required. First inspection in the hills 45 days after planting, in plains 35 days after planting. Second inspection 60 to 70 days after planting. Third inspection immediately after haulm cutting or destruction, and fourth inspection 10 days after haulm cutting or destruction. The fourth is isolation 5 meter for foundation and certified seed. Coming to seed standards. For hill seeds, seed size, the mean length and width of the middle of tuber should be 30 to 60 mm and corresponding weight of 25 to 150 grams, and large size seeds are above 60 mm length and width and 150 grams of weight. In plains, small seeds means 30 to 55 mm length and width or diameter and 25 to 125 gram weight. Large seed means above 55 mm width and 125 grams size or weight. Size is based on mean length of weight. Permissible limit for non-seed size tuber is less than 5%. Cut cracked tuber should be less than 1% by weight. Greenish pigmentation is allowed. The aphid population build up in SUB-TROPICS. In the months of like January to March, the top aphid population is observed, or 52% viral incidents were found in the spring season, whereas autumn and winter crop acquaint with less aphid damage. Different agronomic practices involving in production of quality seed. This is just to summarize which agronomic practices are recommended for potato production. This I have already discussed in the previous slides. The first and foremost is seed treatment with growth regulator like microbial regulators, seed rate, seed size, spacing, method of planting, interculture techniques like earthing up and all, irrigation, fertilizer management, weed management, and haulm cutting. Farm implements and machines used in potato cultivation. This is labor intensive, increased economic viability and timely operations through development of various farm machinery or implements. Potato harvester, which is tractor drawn and manually or engine operated endless screen type potato graders are also used for grading the potatoes, and the other implements like dryers or manual sorters. Potato varieties. The varieties depend on shape of tuber, shape of eye, size of tuber, texture of the peel. Different varieties with specific features. Kufri Baltar, that is medium maturity with large white round oval tubers having flat eyes. Likewise, different varieties are recommended for different area. Gola, Sita, Up to Date, Kufri Kuber, Kufri Handar 1, Great Scott are the popular varieties. The indigenous varieties and exotic varieties grown in India. Indigenous varieties are Agra Red, Chamba Red, Kunur White, Kunur Red, Darjeeling Red Round, Desi, Dhantauri, Gola Type A, Gola Type B, C, Fulva, Fulva Purple, Splashed, Red Long Kidney, Sattu, Shan, Silbilati. Exotic variety Ellie, Aranchausal, Benchausen, Checks Jefficians, Donder Cavalier, Great Scott, Italian White Round, Late Carmen, Majestic, Northern Star, Gregor Cups, Red Rock, Royal Kidney, and Up to Date. The main features of indigenous variety, these are heat, drought, viral tolerant, and other physiological advantages. Exotic varieties are long day adapted only to hill area and prone to stress condition. Here I have listed some of the early maturing, medium maturing, and late maturing varieties. Some of the important early maturing varieties are Sita, Gola, Up to Date, Kufri Kuber, Kufri Kundan, Great Scott, Kufri Naveen, Kufri Neelmani, Kufri Jyoti, Kufri Alankar, Kufri Laukar, Kufri Ashoka, Kufri Nautal, Jawahar, Surya, Khyati. Medium duration Darjeeling Red Round, Kufri Red, Craig Stephens, President, Kufri Sheethman, Kufri Chamatkar, Kufri Sutlej, Kufri Kanchan, Kufri Bahar, Arun, Pushkar, Sada Bahar, Baltar. Late maturing varieties Fulva, Kufri Kisan, Kufri Safed, Kufri Kumar, Neela, Sindhuri, Kufri Jeevan, Deva, Thal Malai, and Kufri Badshah. The different maturity duration are for different purposes, like early maturing varieties are helpful for crop rotation or growing different crops in a year, whereas late maturing varieties give better yield. List of resistant tolerant varieties and their specific features. Here I have listed different varieties and their resistance to different diseases and pests, or many biotic and abiotic factors. Kufri Sindhuri is moderately resistant to early bite, tolerant to PLRV. Other features like it has slow rate of degeneration and more yield under low fertility condition. Kufri Badshah, it has moderate resistance to late blight and early blight, resistant to potato virus X. It is suitable to varied agrochromatic condition. Kufri Khyati is moderate resistant to late blight and early blight, suitable for high cropping density. Kufri Frysona, resistant to late blight and immune to wart. Variety possesses high dry matter, reducing sugars and low phenols. Kufri Bahar is immune to wart, tolerant to Gemini virus. It has low rate of degradation. Kufri Swarna, resistant to early and late blight, cyst nematode, and immune to wart. For South Indian hills, Kufri Swarna is recommended. Kufri Pukraj, this is resistant to early blight, moderately resistant to late blight, and immune to wart. The early bulker and suitable for low input ecosystem. Kufri Chipsona 1, 2, 3, 4 are late blight resistant, and they have good growing conditions for spring season, frost tolerant, and some are resistant to phenols. Kufri Kanchan, resistant to late blight and immune to wart. It has also slow rate of degradation. Then Kufri Jyoti, resistant to late blight. Kufri Jeevan, high degree of field resistant to blight. Kufri Arun, moderate resistant to late blight. Pushkar, resistant to early blight. Kufri Himsona, for late blight. Sada Bahar, moderate resistant to late blight. Kufri Girdhari, high level field resistant to late blight. Alankar, moderate resistance to late blight. Anand, for late blight, moderately resistant to immune to wart. Likewise, there are many other varieties like Kufri Lalima, Kufri Kundan, Kumar, Kuber, Meghna, Muthu, Naveen, Neela. Coming to research institutes in India which are working on potato, Central Potato Research Institute located at Shimla. It was established in 1949. There are 7 regional research stations of CPRI. One is CPRIC Modipuram, located in Meerut, Uttar Pradesh. CPRS Jalandhar in Punjab. CPRS Gwalior in Madhya Pradesh. CPRS Patna in Bihar. CPRS Kufri Fugu in Shimla. CPRS Udagamandalam in Tamil Nadu. CPRS Shillong in Meghalaya. All India Coordinated Research Project on Potatoes, started in 1970, has headquarters at CPRI, and it has nationwide network of 25 centers, including 7 CPRI based centers, 17 state agriculture university based centers, and 1 voluntary center. The residual facts about potato: it needs seed treatment with gibberellin and thiourea, which helps in breaking the dormancy. Seed size of 30 to 40 gram gives better or higher yield for seed size tubers. 60 into 15 spacing is optimum for higher yield. Haulm cutting at 80 to 90 days of planting. Application of FYM increases seed tuber yield and total yield. Drip irrigation improves water use efficiency. Fertilization through integrated nutrient management gives more return per rupee invested. Chemical weed control helps in large scale seed production. Thank you. Hope you liked my video, and don't subscribe my channel. Thanks once again.